Assalamu alaikum guys and welcome to my YouTube channel Biology Articles and MCQs. Guys, our today's topic of discussion is Z scheme. Guys, if we study the process of photosynthesis, then it is very important to study first of all Z scheme because and this is here shown the simple equation of photosynthesis that carbon dioxide and water react in the presence of sunlight and chlorophyll and will form glucose and oxygen. But it is not that simple. For the detailed process, to study the detailed process of photosynthesis, we have to study the Z scheme. And in the Z scheme, or it is also called light dependent reaction, because here we mainly study the use of the solar energy or solar light that is used in the photosynthesis. And in this all the reactions we will study that how the water is taking part in the process of photosynthesis and how it is producing the oxygen and the ATP and NADPH means ATP means adenosine triphosphate energy and the NADP means nicotinamide adenine dinucleotide, and these ATP and NADH will next take part in the dark reaction, and in the dark reaction they will be the production of glucose. Means you can say that the process of photosynthesis is divided into further two reactions: the light dependent reactions and the light independent or dark reactions. So the light dependent reaction of the photosynthesis is also called a Z scheme, in which we study the reaction of or taking part of the water and the production of oxygen and energy and NADPH. And in the light independent reaction, the second which we call the dark reaction, we will see that how the glucose is produced by the help of the carbon dioxide in the process of photosynthesis. So I hope you understand that which we are going to study in the Z scheme, that or what is the Z scheme. Here you can see that all the reactions of the Z scheme are given. But before starting the Z scheme, I would like to tell you that why it is called Z scheme. Guys, it is called Z scheme just because of its Z shape like structure. Previous in the previous books of literature, this the reaction was drawn in the form of the alphabet, English alphabet Z. But now it is rotate clockwise in the 90-degree direction, and it is now in that shape. And the reason of this rotation was also that in order to differentiate different molecules on the basis of their energy levels. Because the different molecules that are taking part in the Z scheme are of the different energy levels. So the here you can see that the molecules that are at the lowest point drawn at the lowest point are of low energy level, and that are drawn at the highest point are of high energy level. And if we compare the redox potential, then the molecules having high energy level has low redox potential, means in the negative, and the elements will having lower energy level will have the high redox potential, means positive. And what is the difference that the molecules having high and low redox potential? That we will see that the molecules of the lower redox potential will transfer the electrons to the molecules that are present at the high redox potential. So this is the difference, or this is why this scheme is now drawn in that form. So start this scheme. First of all, you are seeing that there is written P680. Guys, this is the photosystem, and photosystem, this is a photosystem 2. Photosystem 2 and that is P700, that is photosystem 1. Guys, both of these photosystems contain the chlorophyll molecules, and they are named as photosystem 2 and photosystem 1 just on the basis of the discovery. And the photosystem 2 is also called P680, means photosystem 680, because the chlorophyll molecule present in the photosystem 2 will absorb the energy of the of 680 nanometer, and the chlorophyll molecule present in the photosystem 1, that is P700, will absorb the energy of 700 nanometer, or you can say that the greater than 680 nanometer. So that is the reason of their naming. So starting the Z's came from the photosystem 2, that is P680. First of all, the light will enter, the photons of light will enter into the photosystem 680, and you know that the photosystems are simply present in the chlorophyll molecule. So guys, this Z scheme is taking place in the thylakoid membrane, and you know that the chlorophyll molecules are also present in the thylakoid membrane, and in the chlorophyll molecule these photosystems are present. So this is a photosystem 680, and when it will this photosystem will absorb the energy of the 680 nanometers, and after absorbing the energy of the 680 nanometers, some of the electrons will excite from this photosystem and will then enter into the primary electron acceptor, and this primary electron acceptor is shown here just because it is of the high energy level, but this primary electronics acceptor is itself present in the reaction center of this photosystem. So there is simply, in order to understand that the primary electron acceptor is present on the high energy state, it is drawn in the here. So the electron will excite and then transferred to the primary electron acceptor of the photosystem 680, and this primary electron acceptor is now going to transfer the electrons to next electron carriers. But before studying the next transport chain, we will here it is because the photosystem 680 has lost the electrons, so there is a creation of now an electron hole here that is need to be filled now. So how it will be filled? That the water molecule that is present in the plant will split, and this splitting will also in the presence of the light, that the light will hit the water molecule and will split or break down this water molecule into oxygen and hydrogen and two electrons, and that electrons will be received by the photosystem 680, and the electron hole of the photosystem 680 will be filled. Here you can see that there is production of oxygen also. This oxygen is a half oxygen atom, and this will combine with another oxygen atom in order to create oxygen molecule or oxygen gas. So here we see that one of the reactant of the photosynthesis, that is water, is used, and one of the product of the photosynthesis, that is oxygen, is produced here. So moving towards the primary electron acceptor of the photosystem 680. Now this primary electron acceptor will transfer the electrons to the pheophytin. And guys, this pheophytin is the chemical compound that serve as the first electron carrier in this electron transport chain. Now this pheophytin will transfer those electrons to the plastoquinone, and the plastoquinone is an integral membrane protein, and this membrane protein is already present in the thylakoid membrane of the chloroplast. Moving from the plastoquinone, the electrons will then move towards the cytochrome complex. And here you can see that the cytochrome complex contains the two cytochrome proteins, and these cytochromes are cytochrome B6 and cytochrome F. Moving from the cytochrome complex, the electron will transfer to the plastocyanin, and this plastocyanin is a copper containing protein that is present in the thylakoid membrane. So guys, here you can see that the pheophytin is at present at the high energy level because it is of high energy level protein, and when the pheophytin transfer the electrons to plastoquinone, from plastoquinone it is transferred to cytochrome, and cytochrome will transfer it to the plastocyanin. There is the decreasing energy level, and due to this transfer of electrons from the increased energy level towards the decreased energy level, there will be the production of the ATP, that is energy, cell energy, that is adenosine triphosphate. So here you can see that there will be the production of first molecule of ATP due to this total electron transport chain. Here is the ATP, it is produced. So the electrons are now moving towards the photosystem 1, and these electrons are transferring from the plastocyanin towards the photosystem 1, that is P700, and it will absorb the light of as you know that it will absorb the light of wavelength of 700 nanometers. But guys, as the plastocyanin is entering into the P700, the P700 will transfer these electrons, and these electron excite towards the primary electron acceptor of P700 here, and again is the creation of an electron hole in the P700, and this hole is again this hole will now be filled by the help of this energy that is coming from the light. So after receiving the light, there will be the excitation of the electron, and the electron hole is now filled in the P700. So after after the filling of the electron hole that was created in the P700 molecule, the P700 electron acceptor of the P700 will now transfer the electrons to the ferredoxin, and the ferredoxin are the guys small proteins that contain iron and sulfur, and these ferredoxin will then transfer the electrons to NADP plus. And this NADP plus will now receive the hydrogen that was produced here in the water molecule. These hydrogens are now received by the NADP plus molecule in the presence of NADP plus reductase enzyme, and this NADP plus molecule will then changed into NADPH after receiving the hydrogens that are coming from the water molecule. So here is the production of the second thing, that is NADPH here. So guys, here you can see that the ATP is also produced and NADPH is also produced here, and these NADPH and ATP will now help in the driving of the dark reactions, which we will discuss in the next video. I hope you understand all the things about the Z scheme. If you like this video, then share this video with your friends. Thank you.